Welcome back to our series of lectures. Today we are talking about part 3 of the history of microbiology. Part 3 involves the studying of the first golden age of microbiology. This is our lecture 15. The lecture is presented by Microbiology, the biological sciences. You can contact us for any questions through either our email or phone contact. Kindly subscribe to our channel to get more. First golden age. This is the period between 1857 to 1914, and rapid advances spearheaded mainly by Pasteur and Robert Koch led to the establishment of microbiology. Discoveries included both the agents of several diseases and the role of immunity in preventing and treating diseases. During this productive period, microbiology studied the chemical activities of microorganisms, improved the technique for performing microscopy and culturing microorganisms, and developed vaccines and surgical techniques. Fermentation. In the 19th century, a group of French merchants asked Pasteur to find out why wine and beer soured. One of the key steps that established the relationship between microorganisms and disease occurred. They wanted to develop a method that would prevent spoilage when the beverage was shipped a long distance. At the time, many scientists believed that air converted the sugars in these fluids into alcohol. In 1857, Pasteur found that microorganisms converted sugars to alcohol in the absence of air fermentation. Fermentation is used to make wine and beer today. Souring and spoilage are used by different bacteria. With the presence of air, bacteria change the alcohol in vinegar, that is acetic acid. Pasteur's solution to the spoilage problem was to heat the beer and wine just enough to kill most of the bacteria that caused the spoilage. That's a process called pasteurization. Pasteurization is now used to reduce spoilage and kill potentially harmful bacteria in milk and other beverages, including alcoholic beverages. In 1897, a German scientist, Edward Buchner, who lived between 1860 and 1917, resurrected the chemical explanation of the fermentation. He showed that the process could not require living cells by illustrating the presence of enzymes. Cells produce proteins that promote chemical reactions. This led to the origin of biochemistry and study of metabolism. Effective treatment of many diseases were discovered by trial and error before the time of Pasteur, but the cause of the disease were unknown. The Germ Theory of Disease. Discovery that the yeast play a crucial role in fermentation was the first link between the activity of microorganisms and physical and chemical changes in organic materials. This showed the possibility of microorganisms having similar relationships with plants and animals, or specifically that they might cause diseases. This was known as the Germ Theory of Disease. The Germ Theory of Disease met the resistance at first. For centuries, diseases were believed to be punishment for individuals, crimes, or misdeeds. When the entire village became ill, people often believed that or blamed the disease on demons appearing as foul odor from sewerage or on poisonous vinegar from swamps. Most people in the time of Pasteur found it inconceivable that microorganisms could travel through the air to infect plants and animals or remain on clothing and bedding to be transmitted from one person to another. Despite these doubts, scientists gradually accumulated the information needed to support the need in the recent germ. In 1865, Pasteur was called upon to help fight silkworm disease, which was ruining the silk industry in Europe. Decades earlier, amateur microbiologist Agostino Bassi had proved that another cell-form disease was caused by a fungus. Using data provided by Bassi, Pasteur found that the more recent infections was caused by a protozoan, and he developed a method for recognizing afflicted cells. In the 1860s, Joseph Lister, an English surgeon, applied the germ theory to medical procedures. Lister was aware that in the 1840s the Hungarian physician Ignaz Semmelweis had demonstrated that physicians routinely transmitted infection from one obstetrical patient to another since they did not disinfect their hands. Lister had also heard of Pasteur's work concerning microorganisms to animal diseases. Disinfectants were not used, but Lister knew that phenol, that is carbolic acid, kills bacteria, so he began treating surgical wounds with a phenol solution. The practice saw reduced infections and death, but other surgeons quickly adopted it. His findings proved that microorganisms cause surgical wounds and infections. The first proof that bacteria actually cause disease came from Robert Koch in 1876. Koch, a German physician, was Pasteur's rival in the race to discover the cause of anthrax, a disease that was destroying cattle and sheep in Europe. Koch discovered rod-shaped bacteria, now known as Bacillus anthracis, in the blood of cattle that had died of anthrax. He cultured the bacteria on nutrients and then injected samples of the culture into healthy animals. Koch's postulate states as follows: the suspected pathogen must be present in all cases of disease and absent from healthy organisms. The suspected pathogen must be isolated and grown in pure cultures. The cells from the pure cultures of the suspected pathogen must cause a disease in a healthy organism and experimental animal. The suspected pathogen must be isolated from the experimental animal and shown Koch using microscopy, regional staining, pure culture isolation, an animal model, grew colonies of Mycobacterium tuberculosis on solid medium containing blood serum in 1882. He also later discovered the causative agent of cholera, that's Vibrio cholerae. He developed vaccines for anthrax, that is fall cholera and rabies. This development led to the first administration of rabies vaccine to humans. A young French boy, Joseph Meister, bitten by a rabbit dog using microscopy, July 1885. Pure culture isolation, an animal model, grew colonies of Mycobacterium tuberculosis on solid medium containing blood serum in 1882. He also later discovered the causative agent of cholera, that's Vibrio cholerae. He developed vaccines for anthrax, that is fall cholera and rabies. This development led to the first administration of rabies vaccine to humans. A young French boy, Joseph Meister, bitten by a rabbit dog in July 1885. Vaccination. About 70 years before Koch established that Bacillus anthracis caused anthrax, Edward Jenner, a young British physician, embarked on an experiment to find a way to protect people from smallpox. The disease periodically swept through Europe, killing thousands, and it wiped out 90% of the Native Americans on the East Coast when European settlers first brought the infection to the New York world. When a young milkmaid informed Jenner that she could get smallpox because she already had been sick from cowpox, a much milder disease, he decided to put the girl's story to the test. First, Jenner collected scraping from cowpox blisters. Then he inoculated a healthy 8-year-old volunteer with the cowpox material by scraping the child's arm with a pox-contaminated needle. The scratch turned into a raised bump. In a few days, the volunteer became mildly sick but recovered and never again encountered either cowpox or smallpox. The protection from disease provided by vaccination is called immunity. Pasteur discovered why vaccination worked years after Jenner's experiments. He found that the bacterium that caused cholera lost its virulence after it was grown in the laboratory for long periods. However, the microorganisms with reduced virulence was able to induce immunity against subsequent infections by its virulent counterparts. The discovery of this phenomenon provided an idea of Jenner's successful experiment with cowpox. Both cowpox and smallpox were caused by viruses. Cowpox virus is so closely related to the smallpox virus that it could induce immunity to both viruses. Pasteur used the term vaccine for culture of avirulent microorganisms used for preventive inoculation. The Latin word vacca means cow. Thus the term vaccine honored Jenner's earlier cowpox inoculation work. Jenner's experiment was actually not the first time a living viral agent, the cowpox virus, was used to produce immunity. From 1500s, physicians in China immunized patients from smallpox by removing scales from drying pustules of a person suffering from a mild case of smallpox, grinding the scales to fine powder, and inserting the powder into the nostrils of the person to protect them. Some vaccines are still produced from avirulent and microbial strains that stimulate immunity to the related virulent strains. Other vaccines are made from killed virulent microorganisms, from isolated components of virulent microorganisms, or by genetic engineering techniques. Thank you for viewing our lecture. The next lecture will be on the second golden age of microbiology. Some success at the panel to get more of tutorials at least as golden. The lecture is presented by Microbiology Biological Sciences. You can kindly contact us at any time so that we may give you any of the other tutorials that you may be interested in.